Good morning, students. Today we discussed about the control of flowering. Most people look forward to the spring season and the profusion of flowers it brings. Many vacations carefully time their travels to coincide with specific blooming seasons. Citrus along Blossom Trail in Southern California, tulips in Holland, in Washington, D.C., and Imonis. As spring progresses into summer, summer into fall, and fall into winter, wildflowers bloom at their appointed times. Plants have evolved a remarkable diversity of complex floral structures to attract different pollinators. Flowering at the correct time of years is crucial for the reproductive fitness of the plant. Plants that are cross-pollinated must flower in synchronomy with themselves as well as with their pollinators at the time of the year that is optimal for seed set. Although the strong correlation between flowering and season is common knowledge, floral meristems and floral organ development. Floral meristems can usually be distinguished from vegetative meristems by their larger size. And the vegetative meristem, the cells of the central zone, complete their division cycle slowly. The transition from vegetative to reproductive development is marked by an increase in the frequency of cell divisions within the central zone of the shoot apical meristem. The increase in the size of the meristem is largely a result of the increased division rate of these central cells. Genetic and molecular studies have now identified a network of genes that control floral morphogenesis in Arabidopsis, snapdragon, Antirrhinum, and other species. Floral development in Arabidopsis, which has been studied extensively. The basic morphological changes that occur during the transition from the vegetative to the reproductive phase. Arrangement of the floral organs in four whorls on the meristem and the types of genes that govern the normal pattern of floral development. According to the widely accepted ABC model, the specific locations of floral organs in the flower are regulated by the overlapping expression of three types of floral organ identity. In the case of photoperiodism, a transmissible signal from the leaves referred to as the floral stimulus is translocated to the shoot apical meristem. The plant apex must be competent to respond to this positive signal. Certain plants go through a juvenile phase during which the meristem may not be competent to flower. Another factor can be a requirement for vernalization. These are some examples of plant integrating some endogenous and environmental signals to flower at the correct time. Floral meristems and organ development. Floral meristems can usually be distinguished from vegetative meristems by their larger size. In the vegetative meristem, the cells of the central zone complete their division cycle slowly. The transition from vegetative to reproductive development is marked by an increase in the frequency of cell divisions within the central zone of the shoot apical meristem. The increase in the size of the meristem is largely a result of the increased division rate of these central cells. Genetic and molecular studies have now identified a network of genes that control floral morphogenesis in Arabidopsis and Snapdragon and other species. The shoot apical meristem in Arabidopsis changes with development. Here in the diagram, it's a diagram of plant rosette leaf. This is a primary inflorescence, column leaf, flower. It's a primary inflorescence, and here is the secondary inflorescence. During the vegetative phase of growth, the Arabidopsis apical meristem produces leaves with very short internodes, resulting in a basal rosette of leaves. When reproductive development is initiated, the vegetative meristem is transformed into the primary inflorescence meristem. The primary inflorescence meristem produces an elongated inflorescence axis bearing two types of lateral organs. First is stem-borne leaves, or we can say inflorescence, and other is flowers. The axillary buds of the stem-borne leaves developed into secondary inflorescence meristems, and their activity repeats the pattern of development of the primary inflorescence meristem in the Arabidopsis. Here we see in the diagram primary inflorescence and the secondary inflorescence. Primary inflorescence, actually secondary inflorescence is developed from the stem-borne leaves, or leaves which are born from the stems, and the cornel leaves are arranged at the below of secondary inflorescence in plants. So thank you, students.